Hello student. Now we study about different biological tools required for transformation of recombinant DNA. So biological tools used for transformation of recombinant DNA, they are of three types. The first one, enzyme. Second, cloning vector. It is also called vehicle DNA. And third one, host, that is cloning organism. Enzyme, first example, that is enzyme. The example of enzyme, that is lysosome, nuclease, DNA ligase, DNA polymerase and reverse transcriptase. So nuclease, they cut phosphodiester bond of polynucleotide chain and classify into two types, exonucleates and endonucleates. So exonucleates, they cut nucleotide from end of DNA strands, while endonucleates, they cut DNA within. So enzyme, that is restriction endonuclease or restriction enzyme, characteristic, they are molecular scissor which recognize and cut phosphodiester backbone of DNA on both strands. Second character, 4 to 8 nucleotide long site recognized by them are called recognition sequence or recognition site. Types of restriction enzyme. So restriction enzymes, they are of three types. Type first, type second and type third. So type first, they function simultaneously as endonuclease and methylase. Example, ECOKI, E. coli. Type second, they have separate cleavaging and methylation activity. Example, ECORI, BG second. They cut DNA at specific sites within palindrome. So palindrome is DNA sequence which when read on opposite strand of DNA, it read same. For example, AGCT in opposite direction, AGCT. So these are palindrome. Next, that is type third. So type third, they cut DNA at specific non-palindrome sequence. Example, HPA first and BMBO second. Restriction cutting may result in DNA fragments with blunt end or sticky end. So this table, it shows source of recognition sequence and restriction enzyme. So first restriction enzyme, ALU first. Source of organism, Arthobacterium, lettuce. Recognition sequence, AGCT, opposite AGCT. Product, AGCT, TCGA. The end product is blunt end. Parallel. Second, BAM H First, so bacillus, that is, it is type of bacteria, and recognition sequence GGATCC. It read in opposite GGATCC. So end part, that is sticky end. Third, ECORI, Escherichia coli, RI. So Escherichia coli RY13. Recognition sequence GAATTC, while in opposite direction GAATTC. G for guanine, A for adenine, T thymine, C cytosine. So it also shows sticky end. Last one, HRND in second. So H influenza RD. Recognition sequence GTC GAC, in opposite direction GTC GAC. So these are the product and it show blunt end. So these are different biological tools required for transformation of recombinant DNA.